بدأت أوقف تاني ولا إيه؟ هتبدأ.
Eric has an MD Ph.D. from Washington University in St. Louis, and he pursued a residency in clinical pathology and postdoctoral training in genomics. After that, he was appointed an assistant professor at Washington University. And in 1994, he moved to the intramural program, and is the research program on the NIH campus in the National Human Genome Research Institute. And he's now chief of the Genome Technology Branch, director of the NIH Sequencing Center, and also appointed scientific director and director of the Division of Intramural Research of NHGRI, which is what I just said, the National Human Research Institute. But more importantly, after all of that, I met Eric at the Information Systems and Molecular Biology International meeting in Glasgow this summer, and I was extremely impressed. He gave a keynote address, and it was so clear and so compelling that I decided that it would be really important to get him to ISO. And he has spent the day with engineers and a sprinkling of biologists, but he has been really remarkable at being able to communicate across a huge divide. So I urge you, during his talk, if you have questions, he's gotten very used to people asking him what things mean. And feel free to interrupt him and ask him.
Well, thank you. Pleasure to be here. I guess I've driven from about, not that far to count, about 10 o'clock on the Beltway to about 1 o'clock on the Beltway, something like that. It's a little crazy. I've been here more often. It's really not a very far distance. But it's fun to be here. It's always fun to talk to engineering experts. Indeed, I can't emphasize enough. I was telling people at lunch what a vital role engineers and mathematicians and computer scientists play in the Genome Project. So this is not a foreign concept to have these sorts of interactions. So needless to say, this is truly a spectacular time to be doing human genetics research, evolutionary biology research, and numerous other areas in biomedicine. And this is particularly in light of the recent completion of the Human Genome Project and developments in genetics and genomics. Now, to set a context, really the foundation for this was provided by this landmark publication that took place just about 52 years ago, written by Watson and Crick, reporting the double helical structure of DNA. This is arguably probably the most significant publication of the last century in biomedical research literature. Now, in the intervening 50-plus years, we have seen major advances in our fundamental understanding about the mechanisms by which DNA encodes information that's necessary for all life forms and processes. We've seen the molecular biology revolution come and bring with it unbelievably powerful tools for manipulating DNA, and doing so and being applied in ways of ever-increasing complexity. And we've also seen the successful conceptualization and execution of the Human Genome Project, which of course led to just almost precisely 50 years later, a completion of the human genome sequence. And that's shown here actually is a cover of a DVD, which we actually now distribute, which remarkably enough, it really sets the context for things. You can have on one DVD the entire sequence of the human genome burned on it. And so when you sort of pause for a minute and think about that, in a short 50 years, we went from having the most basic information about our genetic material to actually having DVDs available for distribution that contains the 3 billion letters that constitute our own genetic blueprint. That 50 years, it truly represents a remarkably productive period of biomedical research. Now, at the same time, thinking about what a marvelous opportunity this is, and what the plentiful and really exciting opportunities that all of this creates, I have to be quite candid with you and tell you at the same time it actually is all quite daunting. And if you didn't quite appreciate how daunting all of this is, I think it's important for engineers every once in a while to actually stare at some DNA sequence. So shown here is 1/10,000 of 1% of the human genome sequence. And you can immediately recognize that simple visual inspection fails to reveal the important information that it encodes. And indeed, our ability to actually interpret this sequence is remarkably underdeveloped at the present time. And so really the next great challenge in genomics, which is what I'm here to tell you about, really involves processes that involve experimental and computational work that will allow us to identify all the sequences in the human genome that are functionally important. And in essence, this will involve using various methodologies that basically allow us to go through and, if you will, highlight those parts of the human genome sequence that actually confer function. And at first, what we do need to do is to identify those sequences that actually directly code for protein, otherwise known as coding sequences or protein coding sequences. But it actually turns out that we then need to go on and we need to identify what is actually going to turn out to be a larger amount of sequences shown in pink that actually act as regulatory elements to control the temporal and the spatial expression of these genes, that mediate chromosome dynamics, or that function really in as yet undiscovered ways. And indeed, it is almost for certain that perhaps most, or certainly many, but I actually think perhaps most, of this pink highlighted non-coding functional sequence really reflect elements that aren't even described in textbooks today. That this is actually new ways that DNA can confer function. And we first have to discover these elements. We have to characterize those elements, and eventually we need to catalog all those elements. And so you can see that while there is certainly a handful of tools to do some of these things right now, what I really want to tell you is that indeed I think most of these tools are undeveloped at the present time, and certainly not going to tackle the kind of challenge to fully catalog all these sequences, in particular non-coding sequences. But there is one tool that is actually quite powerful, and probably the one that's being exploited to the greatest degree at present. And that involves using clues that have been left behind by really millions of years of evolution and evolutionary processes that have led to the current human lineage, that being the sequence that's present on a DVD such as this. Well, a real legendary figure in evolutionary biology was Charles Darwin, and among the many brilliant things that Darwin said was that it is not the strongest of the species that survives, nor the most intelligent that survives. It's the one that is most adaptable to change. And indeed, a critical concept of evolution has been that there's continual tinkering that goes on in our genomes. And this is sort of a constant attempt for species to be able to adapt to their changing environments. And through this trial and error tinkering that has gone on, in essence, the non-essential parts of the genome basically get changed in ways that sort of don't matter, and that leaves behind information. But what does happen is that the functional parts of the genome tend to change at a much slower rate, because there's sort of biological processes that are fundamental, and there's only a certain number of ways that you can code for that in our DNA sequence, and so that tends to be the more slowly evolving parts of our genome. And thus, left behind in every animal's genome, if you will, are all sorts of clues about what changes have and have not worked, and what things are allowed to change as organisms are adapted to their environment, and those things that need to be kept more constant. And buried in those clues will allow you to try to get some information about what is and is not functionally important in the genome. And so if you fast forward to a more contemporary genome scientist, is certainly a good friend of mine, Eric Lander, I thought, said it well when he said for the last 3.5 billion years, evolution has been taking notes. And in essence, evolution has left behind sort of this legacy of successful and failed experiments, and all scripted in the genome sequences of current life forms. And by reviewing those experimental notebooks, one can get clues about what is and is not functionally important in our genome. And so the research that I want to describe to you now really is aimed at identifying functional elements in our genome by comparing the sequences of different species. And this is based on the notions that sequences found to be common among animals or species that are separated over large evolutionary distances
are more likely to be functionally important. Otherwise, evolution would have stepped in and tinkered in and made it be different. And in other words, by doing so, in essence, you can use the experiments of evolution to decode the human genome. And this involves basically taking corresponding sequences from two different species, these are often referred to as orthologous sequences, and then comparing them computationally most typically, and doing so in a fashion that allows you to basically look to see what is common and what is different, and those sequences that are found to be common, otherwise you can also say that they are conserved sequences, can be cataloged, and those are the ones that you can basically highlight because of the ones that are most likely to be functionally important. Well, another sort of metaphor you can use is saying that comparative genomics is akin to cryptography, whereby clues are sought by recognizing common patterns of DNA sequence. So shown here are two sequences for comparison. In this case, for illustrative purposes, it's a series of letters. It's not actually a DNA sequence. But if these sequences are aligned just right and scrutinized in just the right way, small patches of common sequences can be found that simply reveal where is the good stuff. And it is that sort of cryptography that is sort of the basis for comparative genomics, comparing different genome sequences. Well, what is the good stuff that we want to find buried within our genome? Well, in thinking about functional elements in the human genome, that is the good stuff, I think it's useful to divide it into two very broad categories, because I think our paths towards their identification are actually going to be quite different. Those parts that code for protein, and those functional parts that don't code for protein. So for simplicity, and for bioinformaticians in the audience, I will say, partially to be provocative, I will make the general claim that coding sequences, that is genes, are relatively easy to identify. Now, why is that? Well, to start with, we know what we are looking for. We have something called a genetic code. We understand how DNA sequence translates into protein sequence, and we know the rules. And as a result, we know what we're looking for at a fundamental level. Meanwhile, we have various complementary data sets that are available to us, mostly cDNA sequence, the parts of the genome that have been transcribed, that are then being used for making protein. And we've amassed these very large computer data sets, and that helps us pinpoint and yellow highlight in the genome where the gene sequences are. Because we're pretty smart, because we've got an head start, we know what we're looking for, we can teach computers how to find them. So we have ever-improving computational gene prediction programs. So I will simply say that I am not worried about our ability to catalog all of these human genes. We're well on our way to doing that. Yes, the last little bit will be hard to tidy up, but I don't think that's the grand challenge. I think the grand challenge is cataloging these non-coding functional sequences. And I think these are going to be very hard to identify. Why? Well, let's be frank. We know very little about what we are looking for. Yeah, we know a little bit here, we know a little bit there. We do not have a good handle on all of the ways that DNA sequence can confer function, especially once you get into the non-coding portions of the genome. We have virtually no complementary data sets available to us, any real strength. And meanwhile, because we don't really know what we're looking for, we have very poor computational predictions. We have some that are getting better, but by and large, I think this is where the problem resides. And as a result, I think this is the great challenge. Now, my research program at NIH really aims to develop strategies for using comparative genomics to really help us identify the functional parts of the genome, in particular non-coding functional sequences. And indeed, we think a major role for comparative sequence analysis is going to be for the identification of functionally important non-coding sequences. And so what I want to now do is provide a little more background on the basis for this statement and give you some highlights of the kind of research that is ongoing that I think will help lead to this conclusion being an accurate one. Well, let me set a little bit more landscape here. Today, efforts to generate whole genome vertebrate sequences have been limited to a small, carefully selected group of species. So complementing the complete sequence of the human genome is a very good sequence now of the mouse and the rat and the pufferfish genome. There are rapidly maturing draft sequences of the chicken, the chimpanzee, and the dog genome. And we're getting pretty good sequences coming on board either already or in the near future for the cow, Xenopus, and an opossum laboratory model, Monodelphis, and also Rhesus macaque. Now, as these sequences are generated, bioinformaticians and computational biologists go through and they systematically compare all of these different sequences, trying to catalog what has and has not changed over the tens of millions of years of evolutionary time that separate these various species. Now, the most detailed comparison that has been performed to date have been between the human genome and two different rodent species, that of the mouse and rat. And there really are four findings from these very prominent papers that feature the first comparative studies of these initial mammalian genomes that I think I should really highlight for you because they'll be relevant for our discussion. It turns out that if you take the human genome sequence and you align it, when you try to line up the orthologous regions of either the mouse genome or the rat genome, 40% of it will velcro together. When I mean by align, you can line up the corresponding parts. 40% of it will align. But it turns out that based on some very sophisticated studies reported in both of these papers, it is now seemingly to be a rough estimate that looks pretty good. About 5% of our genome is under evolutionary selection. It's being retained throughout all these evolutionary forces, implying that about 5% of our genome is functionally important. Now, we now believe that something like 1.5% of our genome encodes protein. But that means the remaining 3.5% that is functional must be non-coding. So the challenge becomes that if 40% of this aligns together, but only 5% of it is actually functionally important, we need some way to distill down, let's say, this 40%, which looks similar, to the 5% that is actually functionally important. If there was more evolutionary time between us and rodents, perhaps this would be a lower number, indeed it would be. But at the moment, you have this 5%, you got to figure it out from among that. We're going to figure out this 1.5% I already told you. But how are we going to ever figure out this 3.5% with all the problems that I alluded to earlier? Well, one approach for tackling that challenge is actually to develop methods that allow you to compare sequences not just two at a time, human mouse, human rat, but to compare all of them at once by having more data. Perhaps it brings more power to the analysis. And indeed, it's almost for certain that while we're going to do that, these currently selected species are almost for certain not the perfect ones that one wants to use for maximizing the power of comparative sequence analysis. So, but at the same time, this is expensive stuff. To give you a little bit of a feeling for what this costs, if you go to sequence a mammalian genome today, and you want to sequence the whole thing, it's going to cost you about $40 to $60 million. So you want to be very careful on which genomes you pick to sequence, because each one represents an investment in several tens of millions of dollars. Well, my comparative genomics program at NIH is trying to address questions that are very relevant to these sorts of dilemmas. And it's asking questions such as, what are the best combinations of additional species to sequence for identifying conserved functional elements in the human genome? And once you have such data sets, how can evolutionarily diverse, multi-species sequences be used to identify conserved elements in the human genome? Now, rather than tackling whole genomes, which require a very large sequencing center, my sequencing group at the NIH is more mid-sized scale, what we do is something a little different. We take targeted regions of the genome, just discrete parts of the genome, and we sequence them. But now we can sequence them in many additional species. In essence, we're doing something that is akin to sort of a reconnaissance project. We're going out there and seeing what's life going to be like in some part of the genome when you have 20 species or 30 species worth of sequence. That will create model data sets that allow you to then develop computational methods for distilling down that sequence in a world of having many species sequences for the purpose of identifying functional elements. Well, we are in fact doing this now, and we are doing this across over 150 different targeted regions of the human genome. But today I'm just going to present you data for our flagship target, the major target that we sort of do, and we basically describe the most in detail. And in this particular region, human chromosome 7, it contains these 10 genes, about a 2 megabase interval. It contains a number of genes. The most famous gene you may be familiar with is the gene that, when mutated, causes cystic fibrosis. And if you want more details about this particular region, this was sort of our announcement paper describing this project. It was published in Nature a couple of years ago, and I refer you to that paper. Now, meanwhile, we're generating a sequence of that region in greater than 40 species so far. But the initial set that we got data for that I'm going to share with you is just this initial group. It included a couple of primates in addition to human. It included this group of additional mammals, non-primate mammals. It included one representative bird and 3 different fish species. Well, we have the sequence data that generated from all these species. Now we want to go about it and we want to compare it. And the way we compare it is that we align these sequences and then we look to see the patterns of sequence conservation. And one of the standard tools that we use for this is for comparing orthologous sequences is a tool called Multi-Pit Maker. And Multi-Pit Maker just simply allows you to view the patterns of pairwise alignments between two species. So shown here is a 30,000, a 30 kb interval right in the middle of the CFTR gene. And this happens to be one of the protein coding regions, an exon here and an exon here, and everything in between is non-coding, or otherwise known as an intron. And in this case, we are comparing the human sequence to, in this case, the mouse sequence. And what you can immediately appreciate is something, and then I can tell you that these little black dots, and I'll show you a plot, represent regions where these sequences are velcroing together, where you have an alignment. And what this is showing is something called a percent identity plot, because everywhere where you see one of these little dark areas, it represents a human-mouse sequence alignment, and it's basically showing its position relative to the human reference sequence from left to right, and it's graphing it as the percent identity between that little section of an alignment, from 50% identity to 100% identity. What you immediately appreciate is that we have really high percentage identity sequence alignments at the positions of the exons. Exactly what you'd expect. Exons, protein coding, functional, should be conserved, should be selected for an evolution. That's why you get this nice position here. But also notice that this is reminiscent of what I said earlier, that just under half of this interval of the human genome also aligns with the mouse sequence. That's why you get little black bits of these little pits. It's just under half, about 40% of this interval. Now, some of these alignments actually are at higher percent identity. They're being conserved to the same degree as these coding sequences. But which ones are functionally important? And which ones are just, because evolution hasn't scrambled them up, just looking like they're functionally important? How do you distill down these bunch of alignments from that 40% down to the 5% that's actually functionally important? Well, just comparing human to mouse doesn't do it. But what about if you look at a larger spectrum of species? So analyzing the sequence from a larger set of mammals immediately reveals sort of the different patterns of sequence conservation. We're looking at the identical interval, and now you can see immediately that here, once again, is the mouse. Here you can see the two primate sequences, which show near 90% or greater alignment across most of the interval. Lots of sequence conservation you expect with our closest relatives. If you go to these other mammals, you can see a pattern that's more reminiscent of what you see with mouse. Chaos, just lots of, I know some of you study chaos, so I need to be careful what I say when I use that word. But basically, all sorts of sort of clouds of these alignments without anything jumping out at you as being more or less functionally important. That pattern changes when you get out to birds, where you can see virtually no sequence conservation notable in the non-coding portion, and the same with the fish. In fact, the only thing in common among all these vertebrates actually turns out to be the alignments that you consistently see in the two exons, the known functional parts. The question becomes, can you do something to analyze these sequences, especially within the intronic regions of these mammals, in some distilling, filtering kind of a way that would reveal certain sequences that are more consistently conserved across multiple species? Because maybe by having those, you could identify discrete regions that are consistently conserved. And in doing so, that might be the greatest candidates for being those that are functionally important. So in essence, what we want to do is take these sequences from multiple species, compare each of them to a human reference sequence, but now in a multi-species synthetic way, compare them all and try to identify ones that are most highly conserved on a consistent basis, because those would be the best candidates for this. And so we're basically just trying to filter the sequence down to find those sequences that are most consistently conserved. And we refer to these types of highly conserved sequences as multi-species conserved sequences, and from this point forward, I will refer to them as MCSs. So in this 2003 Genome Research article, we report our methods for identifying MCSs, and I'm not going to tell you a lot of details, but central to the MCS detection scheme is to calculate a conservation score for each and every base across an alignment containing all these various species sequences. Now, the specific algorithm that we use takes into account the different divergent rates for each species. And by that I mean that a chicken alignment, when it forms, will contribute more to the conservation score than a baboon alignment. Because we almost always align our DNA sequence with baboon, but we do it very infrequently with chicken. So we weight it a little bit based on its phylogenetic position. And then meanwhile, we have to know what the baseline evolutionary substitution rate is between humans and each species. And for this, we use a little trick that's often used in these studies. we simply calculate this on something known as fourfold degenerate positions. These are positions in our genetic code that while they're encoding the amino acid, they can actually vary. And so they're under mutual evolutionary selection. You can use these to sort of calibrate what is the baseline evolutionary rate between humans and a given species. So we can do that. And in essence, let me show you what I really mean by this in a simplistic way. So we can take, let's say, a particular sequence, and in this case, we're taking a region between that is 84% identical between the human sequence and the cat sequence. Well, it actually turns out that 84% there's chicken sequence, and you can see about 64% identical. But that cat alignment actually contributes very little because the baseline substitution rate between humans and chickens is 83%. So it just barely tucked its nose up above that 83% level. But the chicken contributes great, because its baseline is 48%, significantly, the 64% is much greater than that. That contributes in a big way. And we do this with a lot of fancy mathematics, and we basically do this species by species and come up with a weighted conservation score. Well, here, for example, is when you actually do this across a region. This is now going across an interval of 10 kb. You just go along and you just calculate a conservation score base by base. And you get a whole series of peaks. And you have to ask the question, what's significant and what's not? Do you draw a threshold here and say everything above this is the stuff that we want to look at? Do you draw it down there and say everything above that? Do you draw it somewhere in between? Well, in order to do that, you want to sort of calibrate your threshold by accounting for sequences that you know should be under evolutionary selection, sequences you know are functionally important. And for that, we use coding sequences. And so these are used, we know what the coding sequences are across this target. And so this now depicts the known coding bases and the distribution of their conservation score. We can now compare that to everything else. And this is the distribution we get for the alleged non-coding sequence across this interval, which by and large has a lower conservation score, as you'd expect. Well, since we believe that, at least based on the current estimate, that 5% of the human genome is functionally important, we wanted to find a threshold whereby 5% of the bases would be accounted for in the most highly conserved fashion. And that can be simply depicted by identifying those sequences to the right, those bases with a conservation score to the right of that line would be categorized as the MCSs. Notice that it accounts for virtually all, not quite all, but virtually all of the coding sequences, and you get a little bit of the tail of the non-coding sequences. Now, the good thing is, while it might not grab every single coding base, at the end of the day, it accounts for over 99% of all the coding exons. The exons, the little pieces of sequence that actually contain coding sequences. So we are, by doing this, capturing almost all the exons, and in addition, we're capturing a teeny bit of the non-coding sequences, which is exactly what we're interested in. Well, by applying such a threshold, we can go through and we can take all the sequence in our almost 1.8 megabase region, and the top 5% sequence would correspond to these MCSs. And we've done that. And by doing so, what I can tell you is that you end up detecting about 1,600 MCSs. These average about 60 bases in size, and by design they account for about 5% of the sequence. Now, what is that 5% actually, what is it composed of? About 22% of the most highly conserved sequence is the coding sequence. And I already told you that accounts for about 99% of all the coding exons. We capture, by this method, virtually all the sequence that we absolutely know is functionally important because of the coding sequence. An additional 3.5% is the untranslated region that's often associated with transcripts. It's not surprising we capture some of that. Yeah?
When you're breaking this into 5% chunks, presumably you have a minimum size?
25, I didn't mention, 25 base windows. The way this is window, which gets more sophisticated like that, is absolutely right. Minimum size, 25 bases. Should have said that. And this very small percent, it turns out, corresponds to some sequence which are called ancient repeats. These are sort of repeats that existed in our genomes prior to the divergence of all the mammals. And these are thought to be mutually evolving, non-functional. So this was sort of a negative control for us. We hope that we capture very little, and indeed very little of our MCSs correspond to ancient repeats. The real excitement associated with this slide is here. That about 70% of the most highly conserved sequence, conserved almost the same degree, in some cases more than coding sequence, corresponds to completely unknown sequence, presumably non-coding sequence. And indeed, this is exactly what we're going to be the most interested in studying for the kinds of reasons I alluded to earlier. Well, what do we know about these MCSs in terms of some of their properties? Well, let me tell you a couple things. Number one, it turns out that MCSs are slightly enriched within the introns, embedded within the middle of genes, the non-coding parts of gene units, as opposed to the regions between genes. We know that greater than 90% of our MCSs are single copied in the genome. They're not being found all over the place. They're very discrete, appear once in the human genome. And we also know that these MCSs are slightly enriched for the regions immediately upstream of genes, perhaps implying that they might be playing a role in regulating those genes. Now, what's known about the locations of MCSs relative to known annotating features? Well, let me take you back to that same region I showed you earlier, and now I'll show you where some of the MCSs turned out to be. Well, guess what? MCSs corresponded to both exons. Well, that's no surprise, but I could have told you that. So that wasn't very surprising. There is an MCS right there. It turns out that if you do a little bit of database searching, that MCS turns out to correspond to a rarely expressed alternately spliced exon of the CFTR gene. So it turns out it actually is part of that gene. Sometimes it's spliced in, sometimes it's not. Another couple of MCSs are there and there. Again, you do a little bit of database searching. It turns out that those MCSs correspond to regions of the chromosome that are sensitive to cleavage by an enzyme called DNase 1. These are thought to be regions of the genome where the chromatin structure, the packaging of the chromosome, is actually opened up a little. And it is actually thought that where the chromosome is opening up a little might be regions where transcription factors are binding the DNA and helping to regulate how genes are turned on and off. These are sometimes used as a surrogate for regions that might be are important for regulation of genes. It's interesting that our MCSs overlap a known DNase 1 hypersensitive site. So it turns out there's lots of other MCSs here and the rest of our interval, and the great majority of which we have absolutely no idea what they are doing at the present time. Now, we are indeed pursuing various functional studies in an effort to correlate our MCSs with biological function. And as I showed on the previous slide, there are anecdotal examples of overlaps between MCSs and known functional elements. I can tell you that 63% of the known regulatory elements that exist in this 1.8 megabase interval indeed our MCSs overlap at 63% of them. I can also tell you that if you look at the MCSs, you find clusters of known sequences that are known to be bound by transcription factors. And I can also tell you that some of them do contain sequences that look like they are RNA genes or RNA structural elements, non-coding RNAs. And in work I'm not going to describe to you, we're actually now directly assaying some of our MCSs for enhancer activity. These are sequences that enhance the transcription of genes, and indeed we're finding some very interesting corollaries that do indeed indicate that some of the MCSs almost for certain are enhancers. So I'm not going to describe any of these studies. Instead, I thought what I should pause now and sort of tell you some other things about some of these studies. So I think together, our data that I presented so far indicate that we can use multiple species sequences for identifying these MCSs, and that these MCSs at least correlate with functional elements in studies I didn't really describe to you. So we're now interested in trying to address the issue of how to maximize the detection of MCSs by getting sequences from other species. And almost for certain, the reason we want to do that is those initial 12 species I was describing to you, and almost for certain those species I told you about earlier that the complete sequences available are being generated, those are not the best ones. What we want to do is determine what are the best ones to sequence. And in fact, if you sort of look at sort of evolutionary position, most of the data I've shown you so far represent species from these points on the evolutionary time scale. But indeed, there's a pretty big void here between where there was almost too much sequence alignments with the mammals, and it really dropped off almost to having too little sequence alignment by the time you got off the all the birds. Now, there's not much in this intervening interval, but there are some things. There are marsupials, such as the opossum, and there are those weird egg-laying mammals that we all learned about in fifth grade biology, and the platypus. And so we were very interested to explore what those sequences would provide to us in terms of things like MCS detection. So just to give you a flavor, and this is actually a paper that just came out 2 weeks ago in PNAS, I'm just going to show you a few highlights from this, where we, for the first time, generated a pretty good amount of sequence data that gave us some information about the structure and the conservation of marsupial and monitoring sequences. So if we just go to that same region that I've been showing you several times, we return to it, you can see that, for example, when you go to the opossum, you get a very interesting pattern, and that continues when you go out to a monitoring. a pattern of sequence conservation that, especially in the non-coding region, is significantly less alignments than what you see with these other more classic mammals, but significantly more, very discrete regions of alignments that you absolutely did not see in the birds and you did not see in the fish. And just to tantalize you further, let me remind you what those MCSs were. And you can see, in some cases, a very nice correlation of where there were those MCSs, where we had distilled down. Indeed, possum and platypus were doing quite an effective job at helping us find out. So now, one of the other things that you might ask as you start to explore different parts of the mammalian tree, that maybe some of the different species differ with respect to which MCSs they contain. Indeed, there are reasons why we're not platypuses. And some of it's going to be in the coding region, some of it's going to be in the non-coding region. And so we were very interested to try to catalog, if you will, the presence of MCSs among these different lineages. And so this is what we recorded recently. We just took these major lineages, and we simply took each MCS and cataloged which species had that MCS and which species did not. And the data actually turned out to be perhaps not terribly surprising, but I think not interesting nonetheless. About 52% of the MCSs that we have in this region are present in all of these mammalian species. These represent the most highly conserved sequences in the mammalian lineage. About 17% of the MCSs are present in all the placental mammals, that's these mammals, plus the marsupials, but they were not present in the monitoring. There is an additional 4.5% that you find in all the species, but not one of the marsupials. And there's an additional 4.5% that's present in these species and one of the marsupials. And interestingly and not surprising, there's about 4.5% that's present in all of the placental mammals, but not any of the non-placental mammals. And the stuff in black just represents MCSs that had different combinations that didn't add up to anything significant. So I think what's interesting about this finding is that there is evidence that there is lineage specificity with respect to the repertoire of these MCSs among our genomes. And in fact, I believe they are likely accounted for basically a different catalog of functional elements that different species are going to carry with them, and particularly different lineages are going to carry with them, perhaps, and this is the underlying genetic basis for differences we see among different mammalian lineages. So, we are, of course, interested then in basically cataloging MCSs in the human genome because we believe they'll provide these clues about functional sequences. And as you've seen from the marsupial and monitoring data, indeed, some of these species are very contributory and very helpful for finding MCSs. And so we are interested in basically now asking questions to investigate the relative contribution of different species sequences to MCS detection. In order to do that, we want to use a little bit more quantitative approach and we want to be a little bit more systematic. So in order to do this, I wanted to define a concept and then show you some data. We define what I refer to as a reference set of MCSs. These are MCSs that you detect when you use all available data that you can use, all species with the sequence to use, and you do that to detect an MCS. Now, I should highlight, and I really want to emphasize this, these reference MCSs are surrogates for functional elements. They are not synonymous with functional elements. It's just we have to use them as surrogates because we don't have any region of the human genome where we know what is and is not functionally important. If we want to test this out, we have to use just a reference set as a surrogate. Well, in doing so, I want to tell you, I'm going to show you a series of experiments whereby we generated sequence data across the region I've been describing with all of these species I already told you about. But in addition, we have a whole bunch more. Some of them I've already told you, such as marsupials and monitoring. Some other species that give us representation from different branches of the mammalian tree. We also included chicken and fugu, and I think we also threw in lemur. So it's a total of 16 species in this analysis. And for this, we basically took all the species and we detected a reference set of MCSs. We then went through and detected MCSs using every possible subset of species. The best combination of for every combination of 16 species, the best combination of 15 species, every combination of 14, and so forth. And we basically comprehensively performed with every combination, and then compared each subset to that reference set of MCSs, the best set that we get with all 16 species. What's the kind of data that you come by doing that analysis? Well, this is a reference set of MCS. It's now cataloged for coding sequence, UTRs, HOPs, and non-coding, like I showed you earlier. So this is the perfect answer. That's the maximum number of MCSs. Shown here is the best performing, shown here are each individual species And what you can immediately see is that species differ considerably among how well they are at helping you find FCSs. Shown here, for example, you can see the best single-performing species turns out to be the platypus. That's sort of in line quite a lot with several other mammals that are just slightly lower, and then it trails off considerably. Also notice how some species, such as chicken and over here fugu, the pufferfish, are very effective at finding coding sequences, but some of them are much less effective at finding non-coding sequences that are highly conserved. So that's sort of the performance of individual species. But what about best combinations of species? Because we don't really care about individual species as much. So shown here are the results with the best performing subset of 1 species, 2 species, 3 species, and so forth. And notice how by the time you have about 3 species contributing to this, you basically maximized out pretty much your ability to detect conserved coding sequences. But a very different pattern is seen in terms of non-coding sequences, where you can see that you're still actually getting incremental advances in your ability to detect non-coding conserved sequences up to about 10, 11, 12 species. Well, what are these species that represent the best combinations? Those might provide us clues about what sequences we want to get from various species in the future. Well, I already told you that the best performing single species turned out to be this platypus. But what about the best performing set 2 combination of 2 species? Well, that turns out to be platypus and 1 rodent species, either mouse or rat. What about the best performing subset of 3 species? Well, here it's like you're ordering off a French menu. You want to get 1 rodent species of one of these species, 1 of those species, and you sort of go down and you kind of like appetizer, entree, so forth. So anyway, that gets you your best combination of 3 species and so forth off these various other bars. Well, that's interesting. By the way, how does that happen to compare with the performance of the genomes that have been sequenced so far? Because I already told you that the current set of species that have been sequenced may not be the optimal set. They were picked for other reasons. They weren't picked for MCS detection. Well, it turns out that mouse and rat, which I told you we already have, well, mouse and rat together only get you this number of reference MCS bases. You can see that platypus alone actually outperforms it. Well, what about if you toss in the chicken? Because we now know the sequence of the chicken genome. Well, those 3 species together actually don't even perform as well as platypus and 1 of the rodents. Well, let's throw in dog and cow, because those are coming down the pipeline pretty quick. That combination of 5 species pretty much equals your French menu combination of 3 species. Now, why am I showing this? Is this to imply that my institute, NIH, which is responsible for funding most of the sequence, was wasting its money? Well, no, not at all. There were reasons that were not related to MCS detection for sequencing mouse and rat and cow and dog and chicken. But what I hope this does illustrate is that our data, by capturing these large data sets and then being able to analyze them in different ways, is providing a framework that allows us to answer questions about what it might be the 8th, 9th, 10th, 18th, 20th, and so forth species we want to sequence, with respect to at least one metric, that metric being the ability to actually detect MCSs. So I hope this illustrates to you at least one aspect of what our data set has been used for. But there's another aspect that I want to now tell you about, because I think this is very important as to what's going on in contemporary genomics. Yes?
There's something very similar to that, and not simpler, that's going on in engineering, because reverse engineering programs that they are supposed to design circuits. And which if I write down the simple machine learning forms 0 and 1, you won't have the foggiest idea what circuit they're supposed to design. And what we learn in engineering is that simply clustering that you do across circuits and across the sequences, it's going to end somewhere. There's an S-curve.
Yes.
You learn. So somewhere we have to allow for injection of any functionality that we find, and just completely then this starts again and get better. And this first was done for reverse engineering programs, the software, where people understood very well that it takes a combination of model and clustering that it do. And then we learned that this is also true for reverse engineering design programs for circuits. Are there provisions for this kind of injection of knowledge? Because sooner or later you're going to get one function that we understand.
Right.
And that should really give you a lot of light here.
So it's not anything we're doing, but I think the basic strategy that you're describing is, I think some people, as you're amassing these large data sets, are precisely what they want to be looking at. And to try to sort of almost leapfrog ahead in what you're learning. But it's not exactly what we're doing, but I've heard things like that described, especially in some of the more sophisticated bioinformaticians. We're looking at data like this.
Some notion of models.
Right, right.
and then you do model-based clustering, model-based integration. So a lot. I know this perhaps very early.
So what I've described so far to you has been using very small targeted regions of the genome, a couple megabases, and basically going down and collecting data at very deep sampling of species, and doing that almost as a shortcut to get sort of data sets. Now, there are other shortcuts one might imagine that would facilitate being able to get the kinds of sequence data one might want to have for doing these sorts of analyses. And one thing that our data set would provide us the ability to investigate that. And that would relate to not necessarily getting super high quality sequence data. So let me remind you of something that you probably may not appreciate if you don't sort of live and breathe genome sequencing like I do. All the studies I've described to you so far have been done using reasonably high quality sequence. And by that I mean, in shotgun sequencing, you oversample what you're sequencing. You read the same nucleotide typically 8, 9, 10 times. You just generate hugely redundant sequence data sets, so that when you reassemble them, your data is really nice and clean. And even for us, we even went on to refine it a little bit. But we're very interested to ask questions about what quality of sequence is either desired or required for MCS detection, especially if you already have a few sequences that have pretty good reference sequences of high quality. And what are the trade-offs that you encounter? Should you try to get use more species and use weaker quality, or should you try to invest all your numbers in a smaller number of species and just have much higher quality? Well, in order to do that, we were able to sort of use our data sets to address this issue. And so we've actually investigated these issues with my collaborator Eric Lander and his group, and the paper that's actually going to come out, I think, next week or the week after in PNAS, is specifically look at the ability to take a shortcut, a short-term shortcut, whereby instead of getting really high-quality sequence of genomes, get low redundancy sequence data as a means to try to shortcut the identification of MCSs across the entire human genome. And so for these analyses, and I want to show you just the fundamental and most important data that is basically being reported in this particular paper. And for this, basically, we took these 16 species, and we took really high-quality data that we have for those 16 species across our targeted region, and we looked and identified the reference set of MCSs, sort of the surrogate for functional elements. We then reconstructed sequence data sets that provided lower sequence coverage, sort of half-X sequence coverage, 1X sequence coverage, 2X. This is the number of, this is the amount of data that's generated, how many times you write every nucleotide. And we reconstructed it, and then we took each of those subsets, and we then simply asked how effective was it at identifying the reference set of MCSs. Well, here, if you take all 16 species, and you basically do this analysis, this is the curve that you get. And one thing that you immediately see is that in generating super high quality sequence, which would be out here about 10x redundant sequence, you actually don't need it to identify virtually all your MCSs. Even by 5 or 6x, you're basically getting all of them. Indeed, at about half-x sequence coverage, you're identifying almost half of your MCSs. And by the time you get up to about 3 or 4-fold sequence coverage, you're identifying most of your MCSs, or virtually all of your MCSs. Well, how does that compare to a subset of, maybe you got it off taking it, using 16 species to 8 species? Well, those results are shown here. And what you can see is that overall you don't do as well with 8 species. But you're not just trading off overall, you have X number of sequence reads you're going to generate. And how does it compare? Should you distribute fewer numbers across fewer sequence reads across a larger number of species, or vice versa? Well, you can see that at levels of, such as here and here, half-X coverage of each species, of 16 species, equals the same exact amount of money as generating 1X coverage of 8 species. And basically, it's a wash. It's pretty much equal. By the time you go up to 1X coverage of 16 species, that equals the same amount of sequence investment as 2X coverage of 8 species. It's pretty much a wash. But when you get up here to about 2X coverage of 16 species, you actually do better than if you take the same number of reads and generate 4X coverage of 8 species. And so from this analysis and other analyses that we report in that PNAS paper, there has been basically a decision that NHGRI in particular is going to now invest, and is already investing, in a series of additional vertebrate sequences that are going to be generated not super high quality sequence, but lower redundancy sequence, about 2X coverage of each genome. And so shown here is sort of a phylogenetic tree of these key mammals. In black are the species for which we have very good sequences either generated or soon to be generated. And then here are two additional set of species, first in red and then in blue, whereby 2x lower redundancy sequence will be generated. Already we have these 2x coverages obtained in elephant and armadillo and rabbit. And coming soon to a database near you will be cat and penguin, and then the other ones will follow over the next year or so. Here's what I want to emphasize. The purpose for generating sequences from these species is not about getting high-quality sequence of those species. It's not about the elephant. It's about us. It's basically using these species and getting data from them, taking those sequences in mass, comparing it to the human genome, and exploiting those sequences for finding MCSs in the human genome. And so I want to point that out because it is a deviation from what's been done in genomics before, whereby high-quality sequences generated for the species. And I think this shift, while it's very important, is being used in the short term to accelerate our ability to identify the most highly conserved sequences all across the human genome. Yep, I'm sorry? Sure.
So if I understand you correctly, having more species is far more important than the redundancy.
Correct.
So therefore, I should put my mind to get more species.
You're better off diversifying your portfolio across more species. And when you get up to about 2x, it's sort of 1 to 2x is sort of the range that seems to be out.
So now we know quite a bit about evolution trees and stuff like that.
Yep.
So when I select the species, does it stand the reason that I should pick animals or...
It absolutely does. And described in this paper, it all has to do with the branch length, the size of these branches here. And there's all sorts of sophisticated...
The way evolution happened, the previous species were not destroyed, but things were added.
Yep. Yep. So your choice of species is actually very important, and it all matter of how much new information does that species provide. Absolutely. Absolutely. So, in the last few minutes, I'm going to shift gears, and on the last few minutes, I'm just going to admit something to you. I'm going to admit to you that my studies I've described so far have a flaw. They have a fundamental flaw. And that fundamental flaw is that all these analyses that use this reference set of MCSs is actually imperfect, because we have no idea if these reference set of MCSs are functionally important or not. I admit it, they are a surrogate for functionally important sequences. And the reason I had to do that is because, as I mentioned, there's no region of the human genome that you can go in, any region of any size, even 10 kb, whereby there is definitive knowledge of what is and is not functionally important. If we had any such region, I would have used that as the model. I would have gone through and we could have done a lot of these studies on that region. It is in the spirit of that absence of well-defined, well-characterized regions in the human genome that our institute recently launched a project that's just getting out of the gates, but I want to tell you about it because I think it's one of the most exciting projects going on now in human genomics. The name of the project is the ENCODE project. It stands for Encyclopedia of DNA Elements. It is one of these audacious projects that we love to do in genomics with the goal of compiling a comprehensive encyclopedia of all functional elements in the human genome. Well, yeah, no kidding. But being realistic, instead of focusing on the whole human genome, this project has an initial pilot phase whereby 1% of the genome is going to be studied, and that same 1% will be studied by everybody in the consortium involving about 30 groups worldwide. And the idea is to have all sorts of people come to this consortium, apply multiple methods, computational methods, experimental methods. You can use Ouija boards as far as we're concerned. Any method you have that you think can find functional sequences in that 1%. Apply it, contribute the data, we'll analyze it, everybody will share data, and come up with an answer. There are actually 44 regions. They're distributed all across the human genome, all different chromosomes. I can tell you a little bit about how these regions were picked. So it's 30 megabases, about 44 regions. About 14 of these regions were selected because they were extensively studied already. Well-characterized regions such as the flagship region I was describing around the CFTR gene, the alpha globin locus, the beta globin locus, regions that have been studied historically with tons of experimental data, tons of literature. That's only about half of the 1%. The other half was completely picked at random by a sophisticated computer program that went through and tried to capture all different demographics with respect to gene content and rates of evolution or conservation between human and mouse. So this consortium is now well out of the gates, and if you're interested in following it, there's various websites that you can do so. I don't have any data to report to you. You might not be surprised to hear that my sequencing group is heavily involved now in getting sequence data from many, many species across that 1%. It's a simple extension of what I've already been describing to you. You can also read more about it in a paper, sort of an announcement paper that came out in Science magazine the end of 2004, and I would point you to that. So in summary, and to close, let me just tell you that I think what I've tried to communicate to you is that we have a great challenge in genomics now of interpreting the human sequence, cataloging functional elements with particular need to identify new methodologies and new approaches for finding the non-coding portion of the genome that is functionally important. We've shown that I think multi-species sequence comparison is one viable route for helping us get down the road towards doing that. And certainly detection of MCS, I think, is the kind of scheme that one wants to use. It's not the only reason for sequencing genomes, but it's an important one. And I've shown you how we are using our approach for helping to identify and really decide which of the menagerie of animals that are out there one might want to sequence for the purposes of determining the human sequence. One thing I didn't describe at all, but I think is important, is there was a whole other reason why we want to be probably doing a lot of sequence exploration among the primate lineage. And in particular, the methods I described will not give us clues about the unique functional elements that are constituted within the primate lineage. You need a different strategy to do that, and indeed we want to be doing that in parallel. And some of that will be going on within the Decode project. So I will stop there. I will tell you that lots of people are involved in this project, as you can imagine. I have a personal lab, the NIH Intramural Sequencing Center, which is a sequencing group at NIH that's doing all the sequencing I've described. Lots of external collaborators involved in data analysis. And in particular, I should give credit to a postdoctoral fellow, Elliot Margulies, who you saw was the first author on several papers. He's sort of been instrumental in all this MCS work, and various collaborators in the Eric Landers group have been involved in that last study I told you about. So I will stop there and I'm happy to take any questions. Thank you.
By looking at those concerns from the top, people understand the evolutionary process.
Yeah.
But does that exclude the unique human secret that we can quite different from the others, and that's important to us?
No, it absolutely won't. And in fact, that was the last one. It won't even tell us. It'll tell us the common elements among all, let's say, mammals. Maybe then we could, as I showed you, maybe we'll be able to say, here are the unique elements to placental mammals and leave behind the non-placental mammals. And then eventually maybe we will be able to say things by not methods I described, those elements that are unique to primates, but not to other mammals. And then eventually how to find those that are unique to human, that's going to be the toughest of all. And it's probably not, obviously not going to be by comparative sequence analysis necessarily, although there are ideas on if we could only sequence 100,000 people, we might get some clues about what might be functionally important in human, but that's a whole other kind of study. But no, you're absolutely right. I mean, this is going to have to be done sort of by a whole series of methods.
I'm wondering whether if you increase the stringency of your selection criteria for identifying,
Yes.
you would also do something similar with doing the cross-identifying. If you identify those conserved.
Yes. So, well, we have done that, and you can do that. Of course, that was our reason for sort of, at least for now, and it's just for now kind of thing, is sort of picking a threshold that gives us the top 5%. It's based on this other set of studies that say that 5% is about the right number. That's why, but absolutely, you can tweak it up. And it is interesting, because when you do tweak it up, you've got to be a little careful, because if you tweak it too much, you'll lose some of your coding base. You don't want to be losing those. But I think it's intriguing is when you tweak it up, you still do retain some non-coding stuff. So what is some of the stuff that is conserved non-coding, but it's conserved to a greater degree than even coding sequences, some coding sequences. But yeah, you can tweak all of it. Yeah. 5% is under evolutionary selection. Yeah, far more conserved.
So, but then you should distribution is extremely
Yes. Yeah.
normal, or not normal, but there's no evidence for
So
So if you then re-examine the data, what percentage
but
you are sure of the picture,
But the way that 5% was calculated had no resemblance to what we were doing here. I have to think about how I don't think we could quite reconstruct that with our data set. I don't know if we have enough breadth, so we don't have whole genomes to sort of collect that. But we haven't, the short answer is we haven't redone it. I mean, we haven't redone it, and to my knowledge, I'm not sure anybody has redone it, even when they have whole genome sequences, for example, now with monodelphis or dogwood. I don't know if they've redone that to sort of see as you get more species, does that number change? Because all we've done this on so far, I mean, others have done so far is rodent sequences. I think it's a good question.
Let me go back to my analogy and try to ask a question. So, if I try to place under you from engineering, I write down programs that I use to some of the programs will tell machines what chemistry to do, how to create the building blocks of systems, some of them how to get that. So what we had to do in reverse engineering this kind of program, we had to go increasingly to simpler and simpler building blocks.